Coming to the common autopsy pathology related to the Circle of Willis. The most common one is the presence of cerebral aneurysm. It could be a congenital cerebral aneurysm too. It occurs at the site where the two arteries join to form the Circle of Willis. And this happens due to the absence of tunica media in the arterial wall. It is commonly called as berry aneurysm. As you can see in this image, this is due to the berry shape. It is called as berry aneurysm. And what happens when it ruptures? When it ruptures, it results in severe subarachnoid hemorrhage. So, while doing autopsy, how to detect a berry aneurysm? To find out a berry aneurysm, first we need to dissect out the circle of Willis like this you are seeing in this image. After that, we need to float it in the water. So what happens when you float it in the water? The aneurysmal segment or the aneurysmal part will get ballooned out. This is how we identify the presence of the aneurysm in circle of Willis. Here we can see a saccular aneurysm close to the origin of posterior cerebral artery. In this image also you can see a saccular aneurysm close to the posterior cerebral artery. So this is one of the autopsy finding that we see in Circle of Willis. Another one that we need to look for is the presence of any atheromatous changes in the Circle of Willis. Here in this last image you can see there are atheromatous plaques seen on the walls of Circle of Willis. It can obstruct the lumen, so we need to look for these changes also. In some cases, we come across like that how the aneurysm is treated. The aneurysm is usually treated with putting the metal coil. What is the mechanism? When we place the metal coil within the dilated artery, it will induce a local thrombosis. It will induce a local thrombosis, and thereby diminishes the subsequent risk of hemorrhage. So that is the mechanism of placement of metal coil in this. Next, we are going to see about the fixation of the brain. Why we are doing fixation of the brain? Why we are fixing the brain? To make the brain harden. Hardening of the brain is better for the neuropathological examination. For that, we are doing fixation of the brain. So, what are the types of fixation of the brain? It's of mainly 5 types. The first one is immersion fixation. Second one is perfusion fixation. Third one is combination of both immersion fixation as well as perfusion fixation. Fourth one is microwave fixation, and the last one is the electrochemical fixation. So fixation is of 5 types: immersion fixation, perfusion fixation, combination of both immersion and perfusion fixation, microwave fixation, and electrochemical fixation. We will be seeing one by one. First one is immersion fixation. That is the most common technique that we use in our mortuary. In immersion fixation, what we are doing? We are in this image, second image, you can see we are keeping the brain suspended upside down in a large bucket of 10% formalin. We are using 10% formalin, the normal fixative, for at least 4 to 6 week duration. We need to keep the brain in 4 to 6 week duration. What is the advantage of this? In this techniques, the brain floats over the formalin. So it minimizes the artifact that are produced by the compression of tissue against the wall of the vessel. And also, in this 4 to 6 weeks duration, in every third day we need to change. In the first third day we need to change the formalin liquid also. Then how much volume formalin is needed? Here the volume of formalin in the bucket should be at least 10 times the volume of the brain. That we should keep in mind. That is, at least around 15 liter of formalin should be poured in a bucket, and in that we are keeping the brain with upside down. In fetus's brain fixation, some authors suggest that acetic acid can also be added. What is the advantage of that? Acetic acid. The acetic acid makes the brain firmer in fetal tissues. So this is the most commonly used techniques, that is the immersion fixation. Some author also tells that adding kosher salt. that also minimize the artifact because it helps in floating of the brain. That is about the immersion fixation. The other method, second one is perfusion fixation. What happens in perfusion fixation? Here the brain is perfused with a fixative under pressure through the vertebral artery or the internal carotid artery or the branches of both this. The brain is perfused with a fixative under pressure. It is done if the brain is within the skull. It is done to examine the deep-seated regions of the brain. It is mostly used for immunohistochemistry studies of the brain, and it is contraindicated if there is any suspected emboli in the brain. In that cases, this perfusion fixation is not done. That is all about perfusion fixation. Third one is combination of both, that also are done nowadays. And in microwave fixation, the brain is kept under microwave at a temperature of 45 to 55 degrees Celsius. The brain is kept at a temperature of 45 degrees Celsius to 55 degrees Celsius. And in electrochemical fixation also, electrodes and electrochemical methods are used. These are the types of brain fixation. So after fixing the brain, we need to examine the brain. In this image, you can see after fixation of the brain, what we will do? We need to first cut out the cerebellum along with the brainstem, like as you are seeing in this image. After taking out the cerebellum and the brainstem, we need to separate the brainstem, and we need to dissect the cerebellum and the brainstem, and we need to inspect it. Also, you can see we need to take the sections of the brain. It can be done either in the coronal planes or in the horizontal sections also, and we need to see the structures. We need to study the anatomy of the brain. We need to look for any pathologies, any lesions, any infarct, any hemorrhages. We need to inspect the brain. This we will be learning later in detail.